Okay. So yeah, I'm gonna talk about thiolized yeast. So talk about the science behind the strains that you just tried. Before I launch into that, just a brief bit about my background. I have a bachelor's in microbiology from the University of Illinois. Moved from Champaign to Houston, Texas, where I got a PhD in microbiology molecular genetics from UT Houston. That was actually studying yeast, but more in a human health context, studying molecular chaperones for anybody who knows what those are. Um, and then from there, though, took kind of a detour to law school, also in Houston, where I got my JD, and this was all part of the plan at the time. Um, I had the intent on going on to be a biotech patent attorney after that, because to be a patent attorney, you have to have both the law degree and a technical degree, because you are essentially acting as a go-between the inventor and the patent office. So you take an inventor's description of their invention, put it into a legal language, and try to get and draft a patent application for them. So that was all related, but I found that being a patent attorney was kind of boring. So ended up having a chance conversation with one of my colleagues who was starting a brewery in the Chicago suburbs, and sparked the idea to start Omega Yeast. So that conversation happened at end of 2012, and by early 2013, Omega Yeast was born and on the way. Um, so we are located in both Chicago and St. Louis. So all of our production happens in Chicago. All of our research happens in Chicago. My business partner lives in St. Louis, so that's where and he's kind of more in charge of the business side of things, accounting, marketing, all that stuff. So that happens out of our St. Louis office. We have microbiologists, people that were just home brewers. We had a lot of people from the brewing industry. Just have a wide array of people that work with us. And our goal is to be helpful, creative, and fresh. So an overview of what we're talking about today, talk a little bit about biotransformation and what exactly thiols are, and then talk about the thiolized yeast and the enzymes that are involved in this transformation, as well as some details about how we actually engineered these strains. We are very open with how we're doing what we're doing, so people are comfortable using it. Um, Talk about the thiol precursors in the raw materials. So the precursors that are found in the malt itself, and hops, as we've talked a little bit about already, as well as some newer products you might have seen that are derived from wine grapes. And then we'll wrap up with how to maximize, and then where we're going from here. So biotransformation, generally, it's a term that brewers like to use a lot. I think a lot of times without um, full understanding what that means. Not that we have a full understanding either, but uh, in this context, we're defining it as the transformation of chemical compounds within the living system. Um, but what we mean is compounds that are found in the raw ingredients we're using, and through yeast enzymatic activity get converted to compounds we like, usually with fruity aromas. Why should we care about this? Because it has the potential to create new flavors and new aromas and drive new styles of beer, or at least tweak existing styles into something different. It helps us get an understanding of how all the flavors interact and how they can change during fermentation. And so it's just a more full understanding of all of our ingredients and how those lend themselves to quality. And finally, sustainability. Having increasing biotransformation should give us tools to pull more out of our raw materials, so get, squeeze more juice out of the fruit, and maybe find creative ways to use more accessible ingredients, including local malts that can vary in how much precursor they have. Um, And we've seen examples of why we need to think about sustainability. We've had a couple of years of barley crops that have not been great, and the hop crop, from what I'm hearing this year, is not good. I think it's the Czech hops, for example, so sods, they're down something ridiculous. It's like 30 to 40% below uh, their normal levels. So we're going to find sods hops are going to be wildly expensive coming up because their crop's so bad. And I think I've heard even in Yakima, they seem to be down like 10 or 15% in crop yield this year. So we're going to things like yeast strains that are engineered to get more out of your ingredients could be helpful in mitigating some of those things. So yeast biotransformation is, there are a couple of areas that we already know that this is important. The most well-studied is probably the monoterpene alcohols. So for example, something like geraniol, which is in pretty high abundance in hops, gets converted to beta citronellol through the activity of yeast. And that converts something that has more of a floral aroma into something that has more of a citrus aroma. In addition to that, there are lesser known hop esters that can be converted by hops into, converted by yeast into different esters. But again, that not much known there. And then finally, thiols, which there are some hops that have what we call free thiols. So they're thiols in the form that we can already smell. So Citra and Mosaic, for example, have high amounts of free thiols, and that's part of what gives those hops their unique aromas. But what I'm going to be talking about is precursor forms of these thiols that are not aromatic and rely on yeast activity to be converted into an aromatic form. And we call that those thiolized yeast is kind of the term we use for the yeast that we've changed to give them this ability. So they take precursor thiols that are non-aromatic, and these precursors are found both in hops and in malt. And some of the focus already has been on hops, but actually it's the malt itself that carries most of the precursors. And these precursors can be converted by the thiolized yeast, and we're highlighting a couple here: Star Party and Cosmic Punch. Cosmic Punch was our first thiolized yeast, and these two strains actually work via different enzymes that I'm going to talk about, but both work to release one particular thiol from a precursor form. And the result is tropical aroma. So what are they? They are, minimize this so I can see all of this. They are largely responsible, at least in certain fruits, for tropical and citrus aromas. And they are extremely potent. I'll have some illustrations here that give you an idea of how few thiols you need to smell them. And we're talking in the parts per trillion to be able to smell some of them. For example, 4-MMP is one that's we typically see in Citra, gives blackcurrant and box tree aromas, and its aroma threshold is 1.5 parts per trillion. This is not very much. And this is actually the thiol that if you have it in really, really high abundance is the one that's responsible for, if you have a Citra IPA that smells of cat pee, that's what happens when 4-MMP gets to very excessive levels. If any of these aromas were familiar to you and you happen to drink New Zealand Sauvignon Blanc, you may have noticed that connection, and that is because New Zealand Sauvignon Blanc tends to have very, very high levels of some of these thiols, for example, 4-MMP and 3SH. And that's you can find it in other Sauvignon Blancs, but New Zealand is very known for that. And the grapes they use have very high amounts of these precursors as well. And in the wine context, those precursors can get converted by the yeast into these thiols. Nelson Sauvin and Hallertau Blanc also have some of these thiols, which is again why we associate them with these kind of white wine-like aromas. 3SH is the one that we're going to talk a lot about today. That's the precursor of version of that is abundant in malt and hops, and the threshold for that is about 60 parts per trillion. These thiols are, this is kind of a collection of thiols that you find in different fruits that give you some of the aromas characteristic of those fruits. They are all, what we mean with thiols is that that's a sulfur-based compound, and humans are just very, very sensitive to smelling these aromas. There are other thiols that are more unpleasant that can give some, burnt tire and things like that. So it's just a wide collection of compounds, and a lot of them give us exotic fruit-like character that is pleasant in at certain levels. So this is, I enjoy this illustration to give you a sense of how little you need to be able to smell it. So that's illustrated here by we're imagining an Olympic-sized swimming pool, and then how much volume of the thiol do we have to put in, or these other compounds do we have to put into that pool to be able to smell them? So many esters, for example, or aldehydes like acetaldehyde, you'd have to dump the equivalent of either 5 beers up to 21 barrels worth of beer into that Olympic-sized swimming pool to be able to smell it. So, that's not very much, especially on the low end. But then you can go to something like diacetyl, where it's 2.5 milliliters, up to 5 beers worth of diacetyl dumped into that Olympic-sized swimming pool till it reaches the threshold that we can smell. But when we're talking about thiols, we're down in the range of 2.5 microliters to 2.5 milliliters. So 2.5 microliters, you can imagine it's a drop. I mean, just the tiniest drop you can imagine on your finger is what you have to drop into that Olympic-sized swimming pool, and now humans are equipped to smell that. So you really don't need very much. And this kind of extends that to show you that amount that you'd need of each of these different types of thiols in order to smell it. So 4-MMP being one of the lowest thresholds is 3.75 microliters. So a very, very tiny drop into this Olympic-sized swimming pool, and we can smell it. And 150 microliters for the 3SH. There's an esterified version of 3SH called 3SHA that leans more towards passion fruit, and this one has a lower aromatic threshold. Let's see. So what is happening here chemically is there's an enzyme called a beta lyase that is needed to convert these precursors into the free thiol. So in the precursor form, it's usually bound to a more complex molecule. In this case, it's the amino acid cysteine. So the beta lyase can clip the thiol off of the cysteine, and it releases the free thiol along with pyruvate and ammonia. So malt and hops tend to be loaded with these precursor forms. There are multiple versions of these precursors, so this isn't even all of them, but the one that the yeast enzymes tend to act on are these cysteine versions. So it's the thiol bound to a cysteine molecule, and that is what the enzyme can act on. But there are more complex versions of the precursor, including the thiol bound to glutathione. There's also dipeptide intermediates, but this is kind of the theme, is there's something attached to the thiol, and we have to get rid of that attached molecule and to smell it. In this form, when it is bound to cysteine or glutathione, it is soluble and it is not volatile, so it sticks around in the wort, which is skipping ahead why you can do things like mash hopping to kind of prime the wort to give the yeast more more precursor to work on, because the precursor from those hops is going to survive the whole brewing process and be present and when the yeast is around to be able to release the free thiols. So this has been pretty well studied in the wine world. It's still not even fully understood in the wine world, but certainly more studied. But there is an enzyme in yeast called IRC7 that had been shown to release 3SH and 4-MMP from cysteine versions of these precursors that are naturally present in wine grapes. It's inactivated when there are high levels of nitrogen in particular, so it's repressed by nitrogen. This is one of the reasons why you see this more in wine, why it was more well studied, because wine fermentations tend to have less nitrogen, so there's less repression. But in the beer context, beer is essentially always loaded with nitrogen. So the gene is, IRC7 gene is never turned on in brewing yeast. And that is if they have a functional version, because one of the other problems is that a lot of brewers' yeast tend to have mutations in IRC7 that makes them inactive. So they even if they express the gene, it wouldn't release thiols because the enzyme is broken. But other than that, there really hadn't been many beer studies in it when we'd started to get involved in our studies in this. So our first approach was actually to take wine strains that had been advertised to have high beta lyase activity and ferment beer with them. And when we just did that, we got nothing that we could really notice. Nothing that we could attribute to thiol aromas when we tried to ferment beer wort with wine yeast. And that is if you can even, a lot of wine yeast can't even really fully attenuate wort because they don't, a lot of them can't ferment maltose. But even when we found one that was both maltose positive and advertised as having high beta lyase activity, we still weren't getting anything we sensed as having high thiol output. We also tried crossing wine strains with beer strains, so just making new hybrid yeasts that have ostensibly higher beta lyase activity. And none of the hybrids we created were producing significant thiol aromas. And in retrospect, we just attribute this to that nitrogen regulation. So even though these yeast can release thiols in wine, wine is just a very different medium. It's got low low nitrogen levels that repress that IRC7 gene, and it's just never turned on. So even if it has an active IRC7, we don't see it in wort because the nitrogen level is too high. So what we did next was to simply kind of overcome this negative regulation is to replace the promoter. And for those that don't know, the promoter is the region of a gene that essentially tells it when it should be expressed, when the cell should make the enzyme. And it's that promoter that tells normal yeast that haven't been engineered to not make the IRC7 enzyme when there's abundance of nitrogen around. So what we did was plugged a new promoter in front of that native IRC7 gene in two strains, namely our West Coast 1, which is the Chico strain 004, and our British 5, which you'd also probably know as London 3. So just plugged a constitutive promoter, so a promoter that would make this enzyme all the time, regardless of conditions, in front of the native gene. And when we did that, so you can see the amount of 3SH output in the parental strains, Chico, British 5, both well below threshold. So reminding you the threshold level is 60, so that would be up here. So can't smell thiols in either of these controls. But when we constitutively express the native version of IRC7 from Chico, we could suddenly smell it. So when we ferment just wort, no hops in the lab, we can smell thiols out of this flask because we've gotten, about twofold over the sensory threshold. However, when we did the same thing in British 5, we did not. It didn't really look any different than what we saw in the parental strain. And when we looked more closely at the IRC7 sequence from British 5, we found that it had one of these mutations that's known to basically kill its activity. So we can pump out all the enzyme, IRC7 enzyme we want in 011, but it's not going to do anything because that enzyme is broken. So if we wanted a version of British 5, which people love to use in hazy IPAs, that was able to enhance the thiol output, we were not going to be able to rely on the native version of IRC7. So we were going to have to engineer that. So to engineer our yeast, we use CRISPR, which you probably heard of because it won the Nobel Prize last year. And put most simply, what CRISPR allows us to do, it's a programmed DNA cutting enzyme. Basically, you can tell your program this enzyme to cut wherever you want it to in the genome of choice, and you can also create what we call donor DNA that has a gene of interest that you want to insert in the cell, and that DNA is flanked by DNA that looks like the DNA where you're going to cut. And when you cut that, so showing down here, when you cut that DNA, that is an existential crisis for the yeast because you will die if you don't fix that cut. So what we have done is essentially tricked the yeast to take this DNA that we've introduced into the cell and use it to repair that break that CRISPR made, and in so doing, you end up inserting your gene of choice into the yeast. So this is, in a nutshell, how CRISPR works. It's just a programmed DNA cutting enzyme that allows you to trick the cell into taking up and inserting the DNA that you want. So specifically what we needed to do in this case, since the British 5 has an inactive version of its native IRC7, was to take the IRC7 along with the non-native promoter, so the constitutive promoter that's going to make IRC7 all the time, took that whole thing from the Chico strain, flanked it with DNA near the region of the genome we were going to cut, and insert that gene into British 5. So now we have a version of British 5 that has the IRC7 from Chico that is constitutively made, and that is what we called Cosmic Punch. So this relies on only native yeast DNA. It's essentially a rearrangement of native yeast DNA, one gene from a different strain put into our strain of choice. Our more recent releases, so Star Party, Helio Gazer, and Lunar Crush, rely on a different enzyme, and there are reasons for that. This different enzyme is from a bacteria called S. hominis. It's actually a bacteria that is native to our skin, to human skin, just kind of lives on us as a communal organism. And there is an enzyme in there that can also do the same thiol biotransformation, but it does it much more efficiently, which you smelled tonight. And that gene is called PATB. So just like the IRC7 case, we insert the DNA that has PATB flanked by DNA that looks like the area we're going to cut. We cut that, and it loops it into the cell. Now we have a version of our Chico that has the PATB gene from S. hominis to make Star Party. So what you get then is thiolized brewing yeast that can take these precursor forms of thiols that are present in the raw ingredients you use that have more active beta lyases to release thiols to give you aromas of passion fruit or grapefruit or guava, things like that. So to review, Cosmic Punch relies on the native yeast gene IRC7 to make this biotransformation, while Star Party, Heliogazer, and Lunar Crush rely on the PAT-B enzyme from S. hominis. So the raw materials that actually have these precursors include both malt and hops. So, I mean, a lot of people, we kind of focus in on yeast when it comes to aromatics, but in the case of thiols and thiol release, we would argue that the malt is actually even more important. But both of these, both malt and hops, have multiple versions of these precursors. Cysteine bound, glutamine, cysteine, glutamic acid cysteine bound, cysteine glycine bound, and glutathione bound. It is only the cysteine bound version that the yeast beta lyase acts on. And in malt and hops, it's really only 3SH is the most significant thiol that's present in precursor form. So as I mentioned, there's just a lot. I mean, it's hard to convey how much. If you were to, I mean, even in the beers you're smelling, that's only a portion of the amount of precursor there that has gotten released. There is 100 to 1,000 times the amount of precursor than there is free 3SH in beer. There have been minimal studies done that have looked at the amount of precursor present in malt, and we've seen variation, but it has definitely not been comprehensive. We've talked to some maltsters about looking into this so we can, potentially get this included in spec sheets in the future, because the malt you use has an effect on the amount of precursor available to the yeast. But right now it's hard to advise people on that because we just don't have all that information. But some of the things we do know is that kilning destroys the precursor. So by the time you're at crystal malt, you're really not getting any sort of precursor, so it's really in the base malt. We have actually found that oats and wheat don't have any precursor whatsoever. So another way to cut down on the precursor available to the yeast would be to up your amount of wheat or oats in your grist. 3SH precursor levels increased during the boil. Again, not really known why that is. So malt gives you a very, very large potential for thiols. But before there were thiol releasing yeast, this really wasn't a significant source of 3SH. And that's because there was no active enzyme to release it. So we've done, again, just limited studies in this, and this is something that I really hope some maltsters will take up. But this is just an example of looking at thiol precursor levels in different types of malt. This is just 2-row from RAR that we use as our main malt for growing yeast. And you can see the variation in different types of precursors. The Munich that we had looked at actually had about twofold the glutathione bound 3SH that the 2-row had the most of almost every precursor. Vienna had a good amount. And again, here you can see that evidence of wheat and oats essentially having no appreciable amount of precursor. And this is when we used in this case, this would have been our Heliogazer. So we're just thiolized 011. This is the PAT-B version of 011. That's our most recent release. A reminder of the threshold amount of 3SH needed to have that aromatic effect. For 3SH, it's 60. For 3SHA, it's 4 parts per trillion. If we do the parental strain, where in this case, it did show slightly above threshold, but really not detectable. But when you ferment just 2-row, so again, this doesn't have any hops in it. You get 10,500 parts per trillion 3SH, which is massively above threshold. and 1,170 parts per trillion of 3SHA in 2-row. And we saw less conversion in Vienna and Munich. So you can, and even less in wheat and oats. So you can see here that the malt you use is going to have an effect on the amount of thiol release that you have using these strains. Unfortunately, we just don't have, I can't show you a list of providers that have and their corresponding thiol precursor levels. So it's just, something that hopefully we can get that information over time, or maltsters start to show this so you can make those decisions when you're coming up with a recipe, whether you want to, maximize thiol output or hold it back. So when we do sensory, this is kind of what we get, or what people describe using our heliogazer and just malt. So this does not include any hops whatsoever, compared to the parental version of heliogazer, which is our British 5 strain. You get mild fruitiness from British 5. It is British and does have some estery quality. But you significantly push it toward passion fruit, guava, and tropical notes when you use the thiolized version. And it was most significant in 2-row compared to the Vienna and Munich, which is a little more muted. I have a little drink here. So in hops, it's a similar story. Where we've got multiple versions of these precursors bound to glutathione, cysteine, and dipeptide intermediates. And it's really more focused on the 3SH again. Even in hops, it's mostly 3SH. There's not nearly as much of these other thiol precursors. We saw not noble hops and C hops tend to be high in bound precursors, and we're talking in the parts per billion to parts per million of the precursor forms from these hops. But much like the malt side of things, there hasn't been much in the way of comprehensive analysis of hops to say, which varieties or which growing regions. There's definitely a lot of opportunities for a lot of research here to get a sense of the precursor levels from different hops in different regions. Because again, there was no, big drive to do any of this sort of research before there were yeasts that were available to actually do these conversions. This is basically it right here, that from this paper in 2017 that looked at this list of hops. So this represents the most comprehensive data that's available in the thiol precursors and free thiols from these varieties of hops. And really what you can hone in on here is the thiol precursors for cysteine are high in Calypso, which you saw used earlier in one of the beers. And SAS is very, very high in the glutathione version of 3SH. The 3MH, 3SH are the same thing. So it's variable. You can see easily enough right here that the amount of the precursors present in these hops is variable. What hasn't been done, even within SAS, we saw variation here. There's a twofold variation in the amount of glutathione 3SH. What we don't see here is year to year to see if, from year to year this shifts. We certainly know that the quality of hops can vary from year to year based on, monoterpene alcohols, things like that. But we don't know because people have not looked at year to year differences in thiol precursors for hops or, field to field variation, things like that. We really just don't know that much yet. So knowing that there were a lot of precursors in hops and that they tended to be the more complex version, so the glutathione version, which yeast cannot act on, we had the idea to try mash hopping under the theory that the enzymes available, the proteases available in the mash from barley would help to convert the more complex versions of these thiol precursors to the cysteine bound version, which is the version that yeast can use. So that was the idea behind mash hopping, and we saw that work out when we tried mash hopping with, I don't remember, and I don't have the variety here, but when we included hops in the mash and then measured the amount of released 3SH in the final beer, there was a little bit of a bump in British 5, but we got more thiol output in the mash hopped wort compared to the wort that had no hops. And there was even more thiol output when we had them in the mash versus when we had in the whirlpool. Now, this is on the surface kind of hard to explain, because you would expect that if there was no benefit, if there's benefit to putting them in the mash, you'd see the uptick, and we saw that. But you would have expected the whirlpool to have been, even there's no benefit, to have been at least the same level as wort alone, but instead we saw a decrease. And we've seen this repeatedly, that there's something about whirlpool hops and T90 pellets in particular in the whirlpool that actually kind of suppress the thiol output. We still don't know why that is. It's an area of active research, but we've seen this multiple times. And in this case, this was with Cosmic Punch. We don't necessarily see this effect with Heliogazer, which is also based on British 5, but uses the PAT-B enzyme, which is much, much more active. So the theory we have there is that the PAT-B version of the beta lyase is just so much more active that whatever little bit of suppression you get, whatever this mechanism is not even noticed in the Heliogazer version, because it's just so much more potent. So that's something to keep in mind, because we have kind of told people that you want to limit the amount of whirlpool hops, because you could, with these thiolized yeast, if your goal is more thiol output, and that seems to be true with Cosmic Punch, but isn't necessarily true with Heliogazer. So there are things to consider before you run out and start mash hopping everything. And that is, you do get some IBU contribution from mash hopping. And the kind of rule of thumb there is that 30% of the IBUs that you would have expected from a beginning of boil addition for that amount of hops is what you should expect for mash hopping. So in other words, if your program told you a certain amount of hops was going to give you 10 IBUs, you're going to get from a boil addition, that same addition from in the mash would give you 3 IBUs. So the rate you're going to use then, as you can imagine, if you are getting IBU contribution, is going to depend significantly on the alpha acid content. So higher alpha acid hops are going to give more bitterness. So you might have to temper their use. Since sods tend to be so high in precursor and also happen to be low alpha, it's kind of the ideal hop for mash hopping, because you can be more aggressive in its use without contributing too much bitterness. So the addition rates that we've seen, you can translate this to 5 gallons, or but is about half a pound to 2 pounds per barrel, depending on the hop variety. We don't recommend using expensive aroma hops. I would not be recommending putting Citra or Cascade or Citra or Mosaic or anything like that in the mash, because the old rules still apply. All those other volatile hop aroma compounds are going to be lost through the brewing process. So keep that in mind. Don't be throwing expensive hops in there just to eke out a little more thiol output. And again, most of the thiol precursors are going to be coming from the malt anyway. So another way to bump up the thiol output is with certain grape juices. So this experiment just involved blending in 5% New Zealand Sauvignon Blanc juice into wort, and using Cosmic Punch. And when we did that, we saw a 20% increase in the amount of thiol output, and that is because New Zealand Sauvignon Blanc juice happens to be loaded with 3SH precursor. So it takes just a little bit of addition to give you a 20% increase. This, gives you the opportunity to do some interesting wine beer hybrids. An enterprising Kiwi had come up with the idea right around the time that we released Cosmic Punch to take essentially the waste product from New Zealand Sauvignon Blanc production, the grape skins, and dry them out and pulverize them into a powder as a brewing adjunct to give you thiols. And when we first heard about this, we thought it was pretty amazing timing, because from what we had read, the wine grapes are loaded with a precursor, but without the right yeast to release those precursors, it's not going to do much good to just put grape skins in your wort. So we reached out, and we've been working a lot together to try to explore the potential for this, for Phantasm, which is that dried wine grape product in brewing. And our experiments are definitely bearing out that it brings a lot of precursor to the table. So when we do wort alone, we get 417 parts per trillion release of 3SH with our Cosmic Punch strain, but adding Phantasm, and I believe this is probably at about a 2 pound per barrel equivalent, we doubled the amount of thiol output. So grape skins can be a source of precursor to boost thiol biotransformation. So just to give you some idea on, bringing back our picture of the Olympic-sized swimming pool and how much thiol is released by these different strains. So Cosmic Punch was the first strain released based on the IRC7 native yeast beta lyase, and the thiol output is about 10 times over the sensory threshold. It really results in something that's a little more subtle. So what the two strains you guys tried tonight are not what I would call subtle. Those are both based on the PAT-B enzyme, which puts out far, far more thiols. And to illustrate it, Cosmic Punch is putting out, this is actually kind of backwards, I guess. No, not backwards at all. This is illustrating the amount of thiols that are being added to this pool by the particular strain. So Cosmic Punch is putting in, on the order of microliters worth of thiol into the pool, but still, which is still enough to smell it. Whereas Star Party is taking about a shot glass worth of thiols and dumping it in this pool, which is much more above threshold. So really, you can do different things with these strains. If you want something that's going to be more versatile and just kind of give you a slight enhancement of these notes, Cosmic Punch is good for that. Whereas if you want something that's going to be more obvious and more intense, then you'd lean towards something like Star Party that relies on the PAT-B enzyme. And as I alluded to before, if you want something in between, blending strains is a very viable approach, and you can blend it with the parental strain and have a very stable blend, because they otherwise have the complete same growing characteristics, brewing characteristics. So that blend should remain in whatever ratio you start with, because neither strain has a growth advantage over the other. And we have seen a dose-dependent effect on the amount of thiol output. So if you had a 50-50 blend, you should have 50% of the thiol output that you would have had if it was pure Star Party. So it gives you a lot of opportunity to dial things in. So if your goal is to maximize thiols, the things you could do would be mash hopping. I think you get a little more benefit when you're using something like Cosmic Punch, which can benefit from that boost in precursor. The newer generation thiolized yeast are so efficient at putting out thiols from malt. I definitely would not say that mash hopping is necessary. You don't have to do that to get significant thiol output, but it is something you can try. Um, Alternative hop products in the whirlpool is something else that we've recommended, and that's based on that observation that we saw a slight decrease in thiol output when we did T90 pellets in the whirlpool. But this even goes, beyond um, the type of hop that you use. There's definitely some sort of masking going on. I don't know if in the beers you had tonight this was noticeable, but we've definitely noticed that when we do heavily hopped beers, sometimes that thiol, even though we can measure it in extremely high levels and 5,000 to 10,000 parts per trillion, it becomes much harder to pick out when there's lots of hops there. So there's a lot of weird sensory science going on here that is definitely not fully understood, because it's hard to fathom how, like, when you smell that Mexican lager, how that intense of an aroma can blend in with the hops when you get hops at any IPA levels. But that's what we've seen. So if you were to use something like Heliogazer in your favorite any IPA recipe, I would wager it's not going to be nearly as in your face and apparent as the beers you tried tonight. So I think that's there's a lot of interesting sensory stuff going on there. Keep in mind that the majority of the precursors are coming from the malt. So again, you don't have to mash hop to get the effect from these strains. But this is really kind of limited to barley. We've seen very, very little from oats and wheat. We know there's difference in thiol precursors from base malts. I can't point you towards any in particular that are higher than others, but there is variation out there. So it gives you something you can play with. And then, of course, higher kiln malts have less precursors. So another variable that you can consider to modulate thiol output. Phantasm, I think, is starting to be something I've seen in some homebrew shops. So again, this is going to be a good source of thiol precursor. We've had some interest from some of the fruit providers to look into thiol precursors in there. It's that project that hasn't taken off yet, but we expect to have more information on that someday. Certainly possible that there are certain fruits that are going to have a lot of thiol precursors. Passion fruit and guava obviously already has a lot of free thiols, so not unreasonable to think they've also got a lot of precursors. I think these strains allow you to kind of rethink styles to some degree. Cosmic Punch, I think you can roll into your pre-existing any IPA recipes, and that's just going to kind of enhance some of the tropical top notes to it, but it's not going to take over the beer. If you want those styles to be more prominent in a recipe like that, I would lean towards Heliogazer. But again, because of this, interesting sensory masking thing that we see, even in that context, I don't think Heliogazer is going to take it over, but it's definitely going to be more noticeable. When you use these strains in very, very simple beers, like a blonde ale, is when they're going to be unmistakable. And really, that's when you're going to have to, if you're serving this to people, consider what you're calling it, because it's not going to be like anything they've tried before. And it's definitely not everybody's cup of tea too. There are some people that, perceive these thiols as unpleasant things, and that's okay. That's sulfur compounds humans are very, very sensitive to them, and different people perceive different things. So these are going to be polarizing. I mean, that's okay. This is just another color for brewers to play with and tweak. And so yeah, there are a lot of tricks here. Using mostly barley, mash hopping, minimal late hopping. Those are the things you could do to drive it towards the thiols and make the thiols the focus. And if you load it up with hops, it's going to be kind of blended in a little more. So there's a lot of things I think people are going to be able to play with, and having the beers that you guys had tonight, I think, is a good start to get things that are a little more simple, to really put that focus on those styles and give you a sense of what it is, so that if you use it in these other styles that have more hops, you're going to be able to locate it in there, but it's not going to be as dominant. So we still have plenty more to do in there. We need to do more research on the type of hops. We need to do more research on what's going on with sensory to figure out, how why these things are perceived differently in different types of beer. We need to start exploring more local hops and malt to see the variation in the precursors and how that plays with these strains. And to help you kind of build a local terroir with some of your beers, and also help you support the people that are making these ingredients more close to home. There's a lot of avenues for exploration. Of course, what's next? Lager yeast. I guess I should have changed this slide. The lager yeast already exists. That's Lunar Crush. And then the 011 with Pat B is the Heliogazer. And these studies are still going on. I don't know if these thiol bomb beers are going to be a thing because they are pretty polarizing. Some people like them, some people don't. We've already heard that tonight. But we do continue to do research, and we're very open about this research, and we talk about it at various meetings and whatnot. So we'll continue to do so as we learn more. So the big question in all this, and that we continue to explore, is how do yeast and hop aroma compounds interplay? There's very clearly some extremely complex sensory going on here that needs to continue to be explored. There's still not that much known about yeast biotransformation of some of the esters that hops bring. So finally, I would just say, it's important to experiment and use, tonight, the beers you got to try tonight as a jumping off point to inform how you might use these. We do not have all the answers, and we learn a lot of things when brewers just try things on their own that we hadn't thought of. So I definitely implore you to just, experiment. So with that, I'd like to, here's a picture of a crew at our last Christmas party, and be happy to take any questions anybody has.
I've got a question. You mentioned just a few minutes ago about sulfur and some people being more sensitive to it than others. As an example, I didn't really notice anything with thiolized yeast, but my wife did.
Yep.
And so I was wondering sort of how that sulfur relates to thiols. Or is that a dumb question?
No, I mean, I think there's maybe an uptick in H2S put out by some of these strains, and some people are more sensitive to that than others. We did an experiment the other day that was really, really eye-opening, where we fermented a New Zealand Sauvignon Blanc grape juice, and then cut it back to around 6% carbonated. So essentially made a wine spritzer with our parental British 5 strain, our Star Party, and Heliogazer. And so our R&D brewer brought those over to me. I smelled them. I'm like, okay, it's very obvious, and I really like the thiolized version. It was very focused on the passion fruit, grapefruit, smelled really good. Later that day, we sat down with some commercial examples of what they call light Sauvignon Blancs. And in that tasting, we started with 2 commercial examples and then got to our thiolized version. And my perception of our thiolized version, well, actually, even the perception of the second commercial version, everybody was getting firecracker out of that, which is sulfur aroma out of the commercial version. And then when we got to ours, I was getting even more unpleasant things than that. And this is the same thing I'd tried earlier in the day that I liked a lot. So, I mean, the theory that's kind of running through my head right now is that uh, the 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 earlier versions of the commercial examples that I'd smelled in that tasting had high enough levels of the 3SH already, the good smelling, grapefruit um, and passion fruit thiol, that I got desensitized to that by the time I got to our version, and I was smelling the other sulfur compounds that were being put out at lower levels, um, and that made it a much more unpleasant experience. But it was the same thing I liked earlier in the day. Um, so we've definitely found, just even a context specific, like, not just like one person not liking these aromas. Whatever you had before, whatever you ate, whatever you smelled, whatever you drank, can affect how you're perceiving these things. And this is just, I think, the nature of these sulfur compounds. We're just so sensitive to them. We have a really hard time doing sensory with these really high output strains like heliogazer. If we're trying to say how does dry hopping affect it, like we try the first sample we smell doesn't have dry hops, and we get pummeled by this aroma. By the time I smell the second, third sample, I've lost it. I can't smell anything anymore. Um, and you start to pick up other things. So it's there's some wacky sensory science going on here that we don't fully understand. People are sensitive to sulfur compounds at different levels. So there's a lot going on. We don't have all the answers now, but definitely they're polarizing because some people like them, some people don't. Depends on the context, you're tasting and smelling. It's all a lot of things going on.
Hey, Lance, question here. So first of all, thanks so much for coming out here. This has been a great presentation, real eye-opening. My takeaway that I didn't realize was how important the malt really is to these thiol production, and so that's like a kind of a wow moment for me. My question is that so there's all the focus seems to be on the 3SH thiols. Is that really what it's all about, or is there any need or advantage or desire to try to look at other thiols that are coming out?
Well, yeah, I mean that's what it's about, because that's what's in malt and hops. So there's as far as these precursor forms. So like I mean, if we went back and looked at that chart, there just, I think 4-MMP was one of the other ones they were looking at. There just isn't much at all of those other precursors. Um, if there were cysteine bound versions of them, we would expect the enzymes, the same enzyme, PAP-B and IRC7, to work on those precursors. So it's just that naturally there's just tons and tons of the 3SH. So I mean, if there was another source of those thiols out there, that would be a possibility, that of bringing ingredients like that to beer to be able to release those. But at this point, I'm not aware of things like that.
Thank you.
Mm-hmm.
Hi. Um, so I had a question. Oh, sorry. I had a question, and I don't know if you quite have the answer for it, but I have played around with these kind of yeasts, and I've been told, at least, that maybe the children of these parent yeasts that have high precursor thiols can also produce a high amount as well. Have you guys played with maybe the children of those parent hops that you say have high precursor thiols per se? Yeah, let's go ahead.
So like new version, like the hops that have like Saaz as their parent, things like that.
Yeah.
If I'm not, isn't Cascade? I'm. This is not my area of expertise. I think, if I remember, isn't Cascade at least half Saaz or something like that?
I believe so. I was more so curious on kind of like the cashmere, like the children of Cascade, things like that.
Yeah, and this is something I don't have the answer to. There's
Okay.
just yeah, that research hasn't been done. I mean, this would be something we'd be happy to do ourselves, just send hops off for analysis. But all of those, every chart you saw in here where you saw reading of thiols is a result of us sending a sample to a lab in France that happens to be the best in the world at measuring these things, because they're very, very hard to measure accurately. There's nowhere domestically that we can send these to get that kind of quantitation. So every single sample or every number you saw in there was effectively a $300 sample. So it's really expensive to do all these. We've spent a lot of money getting thiol analyses done by this lab in France. So it's something I wish the hop growers would pay for.
Thank you.
Yeah.
That was a really interesting presentation. One of the things that I took away from your presentation is because these thiols are so potent, it's probably going to create a huge problem with consistency in commercial brewing. And I would assume that that's why you're trying to get, the malt analysis, to include 3SH and other things that would affect these thiols. Is that a correct takeaway from this presentation, and how far have you gotten in getting people to add that to the malt analysis?
We've gotten nowhere in getting them to add that to the malt analysis. We've talked to some of the smaller maltsters that have expressed interest in, kind of surveying things and diving into this, but we haven't gotten to the point of having, data that could be released. It's somewhat frustrating, but hopefully we'll get there someday, and we continue to talk to maltsters to look at this sort of thing. But I mean, it's kind of the same thing where they'd have to send this off to France to get that analysis done, and maybe you're just resistant to spending on that. So it's something I'd like to see done someday, but I haven't gotten there yet.
Thanks for the great presentation. I have a quick question about the practical use of these hops, as far as using them across multiple using across multiple generations, and if you've noticed any difference from the base yeast from which it's derived.
No, I mean, you should be able to they should behave as far as all the other brewing characteristics, flocculation, repitching, all that. You should expect the exact same performance you would from the parent. The genetic change is totally and even thiol output from generation to generation should be only dependent on the precursors available to the yeast. That genetic change is totally stable. That's, I think, one of the advantages of using these kind of existing brewing strains, is that they're tried and true for their brewing characteristics, and we've just kind of given it another trait.
Cool, thank you. Any other questions? So looks like that's all the questions, but Lance, just wanted to say thank you again for an amazing presentation, super informative. Thanks for drilling into the secrets of thiolized yeast. And it was really fun and kind of mind-blowing. So here's cheers to Lance.
Thank you. Yeah, I look so youthful in that picture. Thank you. Thank you. I appreciate the invitation, and I'm stoked that you guys, brewed these beers in advance to be able to have those there. As it's one thing to say they smell like New Zealand Sauvignon Blanc and grapefruit and passion fruit and whatnot. It's another to actually experience that. So it helps
nothing like tasting it for yourself, for sure.
Yeah.
All right. Thank you, sir.
Yep.
Cheers.
Thank you. See you guys later.